everyone, and welcome to today's webinar, CGX10 Cell Isolation System, Introduction, Utility, and Assessment in a GMP-grade Environment for Multi-Parametric Cell Sorting. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Sony Biotechnology. To learn more, visit www.sonybiotechnology.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. There will also be multiple poll questions throughout the webinar. If you could please take a moment to submit your responses when prompted to do so, we would greatly appreciate it. Your first poll question should be showing now, so please submit your responses. I'd like to now welcome our speakers, Jerry Barnhart, Manager, Field Applications, Sony Biotechnology, Inc., and Manoja Eswara, Senior Development Manager, Center for Commercialization of Regenerative Medicine. Jerry, you may now begin your portion of the presentation.
Welcome. I'm Jerry Barnhart, Manager of Field Applications at Sony Biotechnology. We are here to talk about the next generation cell sorter by Sony, the CGX10 Cell Isolation System. In this presentation, we are going to introduce the CGX10, talk about its key features and its utility in a GMP-grade environment. Sony collaborated with the Center for Commercialization of Regenerative Medicine to generate some technical data utilizing the CGX10 in different cell therapy workflows and assessing its utility in GMP-grade environments. Joining me on this presentation is Dr. Manoja Eswara, Senior Manager of Process and Analytical Development at CCRM, who will be presenting her data. This presentation will be divided into two parts. The first part will cover the CGX10 introduction, and the second part will discuss the data generated at CCRM Canada, assessing the utility of the CGX10 in GMP-grade cell therapy manufacturing workflows. Let's get started with the first part of the presentation. Cell therapy is a revolutionary technology, and the field is rapidly evolving. New workflows are producing more potent and effective therapies. Here's an example of a few of these technologies. In these examples, more efficient therapies are developed using immune cell subtypes. To understand this better, let's look at the T-cell differentiation process at the bottom of this slide. The naive T-cell differentiates into memory T-cells and eventually into effector T-cells. Several studies have shown that the more naive the T-cells are, the better the patient prognosis. So cell therapy developers are utilizing the less differentiated immune cell subsets in their manufacturing workflow to develop more potent therapies. To cater to these evolving requirements and advances, we have developed the next generation cell isolation system. Introducing the CGX10 Cell Isolation System, which leverages Sony's legacy in the field of optics, innovation, and automation. The CGX10 Cell Isolation System is a novel system which enables GMP-compliant cell sorting and is intended for cell and gene therapy manufacturing workflows. The instrument has a proprietary microfluidic chip and built-in temperature control cabinet. The CGX10 enables fully closed, high-speed, high-purity, multi-parametric fluorescent-based cell sorting. Some of the key features are a fully enclosed microfluidic chip for cell sorting. The instrument consumables and software are all GMP compliant. Multiple user modes, a compact design and touchscreen user interface. Temperature control cabinet and automatic clog detection and declogging mechanism. And multi-parametric selection to ensure high-speed, high-purity cell sorting. When you purchase the CGX10 system, you receive a beautifully designed instrument and user-friendly software. We also offer all the consumables which goes with the instrument. The bottom picture shows a depiction of our microfluidic device, which offers fully enclosed cell sorting. By introducing the CGX10 Cell Isolation System, we are enabling GMP-compliant cell and gene therapy manufacturing. we are delivering the GMP promise by offering a compact instrument, which is manufactured in an ISO 9001 setting and can offer fully closed cell sorting. The consumables are also manufactured in ISO 13485 setting. The software is 21 CFR Part 11 FDA compliant. The interface is touchscreen, so there is no need for a bulky laptop or monitor, and the software offers multiple user modes. The instrument seamlessly integrates into a typical CAR-T workflow and offers subtype-based cell sorting for developing cell therapies. This is a representation of a typical CAR-T workflow, which starts with a patient sample draw and apheresis. The T cell is then isolated from the apheresis sample. If the workflow requires, the user can sort the cells based on a particular cell subtype. Similarly, after the engineered cells are expanded, the user can select for cells exhibiting a particular subtype to ensure the therapies they are developing are efficient and effective. To understand why this is important, let's look at the figure on the right-hand side. This is a depiction of the stages in the T-cell differentiation process. Numerous studies have shown that naive T cells show more stemness, better survival rate, and higher proliferative potential. They are more persistent and ensure better prognosis for the patient. Whereas if the cells used in therapy are from the right-hand side of the spectrum, they are exhausted and have a poor prognosis. We gave you the example of utilization of the CGX10 in CAR-T workflows, but the instrument is very versatile and can be used for a number of other applications, such as CAR-NK, CAR-Tregs, tumor-infiltrating lymphocytes, and gene therapy workflows such as cells edited with CRISPR-Cas9 technology. I would now like to show you a short video about the CGX10.
The CGX10 Cell Isolation System from Sony is designed to deliver multi-parametric cell sorting in a fully closed format. A key component of the instrument is an innovative microfluidic chip that is integrated into the system consumable kit. In this video, we will introduce you to the process of fully closed cell sorting taking place inside the microfluidic chip. Here is the overview of the fully closed fluidics design of the CGX10 Cell Isolation System. Once the single-use kit is installed on the CGX10 system, the first step is fluidics priming. In this step, the collection buffer is allowed to flow through the system. Next, the sheath buffer automatically flows through the entire system to prevent bubble formation or entrapment and is drained into the waste bag. The alignment of the chip is achieved automatically by the system by looking at an image of the fluidics channel within the microfluidic chip using a vision system. Following sample introduction, the final optimizations will be completed. As the sample is introduced, the microfluidic chip position is adjusted automatically by the system to ensure high optical signal quality. At this point, the system is ready for data collection. As the sample is flowing, the optic system detects the cells of interest, and the sort delay timing and other parameters are adjusted to ensure the collection of cells matches the desired outcome of high purity at high yields. Clogs that may be formed during sample flow are automatically detected through the scatter-based detection of the sorted sample. Once a clog is detected, the system automatically starts breaking the clog by controlling the fluidics, making multiple adjustments as needed. After the system is calibrated, the sample is hydrodynamically sorted into the collection bag based on sorting criteria. Negative pressure is created based on sort calibration parameters, and the sample is aspirated into the sort chamber. Finally, the sorted cells are collected into the collection bag, placed inside the temperature-controlled cabinet. This is how, with the unique microfluidic chip, the CGX10 instrument enables fully closed, high-speed, high-purity, multi-parametric fluorescence-based cell sorting for all kinds of cell therapy manufacturing workflows.
Now I would like to invite Manoja to talk about the CCRM study focusing on utility and assessment of the CGX10 in a GMP-grade environment.
Thank you, Jerry, for the excellent overview of CGX10. I'm here to present some of the work that we have done with CGX10, trying to integrate it into cell and gene therapy manufacturing processes. And today I'm going to talk about how we assessed CGX10 for its GMP readiness. Just to introduce myself, my name is Manoja Eswara. I'm a Senior Development Manager at Center for Commercialization of Regenerative Medicine, CCRM. CCRM is a CDMO. It's situated in Toronto in a unique top-ranking biotechnology ecosystem. We leverage our extensive experience in enabling technologies through collaborative processes and specialized infrastructure to solve big challenges in cell and gene therapy through seamless integration. We have a large facility with process development lab called CAT-CT, which is co-located with our GMP facility named CCVP. Our GMP facility boasts of a QC lab and 10 clean rooms, 8 rooms for cell therapy and 2 for viral vectors. The current facility supports clinical manufacturing, and we are expanding our facilities to provide late-stage clinical and commercial GMP manufacturing as Omnia Bio. CCRM and Sony collaborated to test the utility and integrability of the CGX10 in cell therapy manufacturing processes. In phase 1, we tested the applicability of CGX10 in a few cell therapy workflows, such as immunotherapy and iPSC-derived cell therapy applications in our process development lab. The data generated in phase 2 are available as app notes. In this presentation, I will be focusing on the phase 2, where we assessed the GMP readiness of CGX10 for independent operation in a clean room. GMP is a highly regulated environment, and moving any process, reagent, or an instrument into GMP involves thorough risk assessment and diligence from a facility and a product perspective. For this assessment, we mainly focused on 2 metrics: GMP suitability of the instrument and independent operability in a clean room. To demonstrate the GMP readiness of CGX10, we performed a comprehensive assessment, such as risk assessment for CGX10 instrument and consumables, supply chain and procurement assessment for the materials, followed by assessment for cleaning, transfer, and IQ/OQ of the instrument, environmental monitoring, and finally 21 CFR Part 11 regulation of the software. For the second arm of independent operability, because CGX10 is a flow cytometry-based cell sorting equipment, some basic understanding of flow cytometry is required by the operators to adjust certain sort parameters during execution of the protocol. Therefore, the objective of this task was to demonstrate the feasibility of operating the CGX10 by trained GMP operators independently in a clean room, following protocols and batch records that are developed in the process development lab. So in the first part of this talk, I'll be discussing our assessment of GMP suitability of CGX10 and the results that we obtained. As the first task, we performed a USP 1043 criteria-based risk assessment of raw materials for the CGX10 consumables, including the single-use kit, accessories, and related reagents, and we performed assessment for attributes such as manufacturing QMS, traceability documentation, animal origin, adventitious agent testing, sterility, and endotoxin testing. Based on our assessment of the documentation provided by Sony, CGX10 kits and reagents needed for cell sorting have been deemed acceptable for usage in a GMP clean room. For any clinical application of CGX10 isolated cells, appropriate regulations and product testing must be followed for manufacturing and use of cells in humans. Any material or consumables that is to be procured and transferred into a GMP facility needs to meet certain quality criteria. In this task, CCRM performed vendor qualification of Sony prior to purchasing single-use kits and CGX10 consumables. Upon Sony meeting the CCRM criteria for a qualified vendor, materials were procured and received under QA supervision. CCRM assessed the validity of the COA, COO, and SDS documentation of the received consumables. A thorough assessment of packaging and labeling of CGX10 consumables were performed to assess if they contain the necessary information and that the consumables are within specifications for use in a GMP clean room. Upon confirmation of all these criteria, materials were transferred into the clean room. Any equipment or materials that are introduced into a GMP facility have to undergo appropriate cleaning procedures to minimize the introduction of viable and non-viable particulates and the risk of cross-contamination of the facility. CCRM performed a thorough risk assessment for CGX10 instrument and related tools for their cleaning suitability, following GMP acceptable procedures, and a risk mitigation plan was developed. CGX10 is not compatible with hydrogen peroxide fumigation. However, it is compatible for cleaning with Preempt and 70% IPA, both of which are GMP acceptable cleaning solutions. It has a very easy to clean design, and inaccessible parts such as the drawer underneath can be detached and cleaned. The instrument is compact and on wheels, and it makes it easier to transfer between clean rooms. In the next task, once it is transferred and installed in the clean room, CCRM performed assessment of the vendor IQ/OQ protocol. This is important because all equipment that supports batch manufacture, which have an impact on process parameters and product attributes, must be qualified before they are used for GMP manufacturing. In this task, CCRM performed qualification studies to verify that the CGX10 and installed and operated within established limits and tolerances in accordance with the manufacturer's specifications. Based on these assessments, CGX10 system has been deemed suitable to install and operate in a GMP facility. Next, we performed environmental monitoring to assess the impact of CGX10 on the clean room. CGX10 has 3 fans that may generate particles when in operation. Therefore, we need to assess that CGX10 passes the EU Annex 1 acceptance criteria for particulate counts for GMP use. To study the impact of CGX10 operation on the cleanroom environment, a tailored protocol for environmental monitoring was developed based on the design of the instrument and the location of the 3 fans within CGX10. In addition to the daily in-operation EM, we performed the CGX10 specific EM for 5 runs when CGX10 was in operation. As shown in the pictographic depiction here, the green rectangle is the location of CGX10 in a grade B clean room, and we placed 3 particle counters right adjacent to the CGX10 where the fans are located. To summarize the results from 5 runs where CGX10 was in operation and where particulate counts were measured, we observed that in all the 5 runs, CGX10 passed the EU GMP Annex 1 acceptance criteria for in-operation particulate counts in grade B environment. And when we take a closer look at the exact number of particles, they are less than 10% of the acceptance criteria. These results indicate that the operation of CGX10 in a clean room does not have an impact on the clean room environment. The CGX10 software used to control the CGX10 isolation system should meet FDA 21 CFR Part 11 requirements to be acceptable for GMP manufacturing. The CGX10 software has been built to accommodate these requirements, including user access restrictions, password protection, secure data storage, electronic records, electronic signatures, and audit trail for traceability of operations. These guidelines ensure the authenticity, integrity, and confidentiality of the electronic raw data. As part of the GMP readiness assessment of CGX10, CCRM supported assessment of the software's 21 CFR Part 11 related features through execution of protocol based on Sony's URS. Minor gaps identified were fixed for the final version of the software to meet the regulation guidelines. To summarize this section of GMP suitability assessment of CGX10, we have identified that CGX10 kits and related products are acceptable for usage in a GMP facility and meet the criteria for GMP procurement. CGX10 and related tools can be cleaned following GMP cleaning protocols and with risk mitigation. Sony protocols for IQ and OQ passed the CCRM criteria for GMP instruments. CGX10 operation did not impact the cleanroom environment. And finally, CGX10 software supports 21 CFR Part 11 regulations for electronic raw data. In the next part, I will present on the assessment of independent operability of CGX10 in GMP cleanroom. 3 protocols were developed and executed as part of this assessment: a QC run protocol to perform routine maintenance, broth run protocol to perform sterility assessment, and a T-cell sort protocol to isolate T-cells from frozen leukopaks in a clean room. We integrated multiple GMP suitability assessments of the instrument into these 3 protocols as outlined in this table. We sourced all GMP-grade materials, integrated these protocols into the assessments, and developed well-rounded batch records. We trained operators who have some or no flow cytometry experience, and after thorough training, we created user-specific profiles and locked the protocols on the CGX10 in the process development lab. Once the instrument has been moved into the clean room, these protocols were then executed in the clean room. For the first protocol, we performed a QC run. A routine quality control run of CGX10 is recommended to ensure sorting sensitivity, stability, detection sensitivity, calibration, and fluorescence detection performance. The QC protocol will need to be performed by trained operators in a clean room as needed in instances such as for routine maintenance, movement of CGX10 between clean rooms, instrument performance assessment over time, and to standardize between instruments and team members. We developed a protocol and batch record for the QC run, and trained operators could successfully perform this QC run by following the batch records with no deviations. Moreover, CGX10 generates signed QC run reports to facilitate documentation. The second protocol that we developed and executed was that of a broth run. Any new process in a GMP environment has to be assessed for sterility risk by performing broth runs or aseptic processing runs. As outlined here, execution of sorting protocols using CGX10 involves multiple welding steps to connect the several components of the single unit kit. Moreover, there is a part of kit installation, sample transfer, and in the event of any errors, the error recovery involves a number of handling steps. All these steps lead to sterility risk of the final product. So we developed a customized broth run protocol to simulate the sorting process, including high-risk welding steps, as well as commonly encountered error recovery simulation steps. As outlined in the figure, all input buffers and sample bags have been replaced with tryptic soy broth and connected to the CGX10 single unit kit. As a control, unopened input TSB samples were tested for growth promotion according to USP 61 guidelines. Sterility testing was then performed on the collection bag contents before and after error recovery tests, and these tests were performed according to the USP 71 guidelines. To summarize the results from 3 independent broth runs, CGX10 processed samples, both the in-process sample and the final sample, passed the USP 71 sterility testing. These results indicate that the CGX10 and the single-use kit operation steps per se do not pose a risk to the sterility of the processed sample. We highly recommend process-specific aseptic processing runs for every new process and sterility testing of the final product as mandated by regulations. For our third protocol, we tested the independent execution of a cell sorting protocol using CGX10 in a clean room. For this part, we chose to isolate naive T-cells from a leukopak as our proof of concept protocol. We chose this T-cell subset because there are studies to support that naive T-cells have increased persistence in vivo. The therapeutic objective of using this subset of T-cells would be needing to infuse less cells into the patient since you are infusing cells that have a longer efficacy. Currently, there are no commercial reagents available to sort this subset of cells that require multiple markers to differentiate from other T-cell subsets. For our studies, we stained the cells with a combination of CD4, CD62L, and CD45RA to isolate CD4 naive T cells. To go into the details of this process workflow, a frozen GMP leukopak was thawed on day -1, and RBC lysis was performed to remove red blood cells. Cells were rested overnight at 37 degrees. On day 0, 500 million cells were stained with a cocktail of fluorescent GMP antibodies for CD4, CD45RA, and CD62L. Cells were sorted using a purity mode for 4 hours. At the end of the sorting, sort purity was checked on the pre-sort and post-sort samples using Cytoflex. Viability assessment was performed using NC200. Once the protocol was developed in the process development lab, it has been locked so that manipulations can be minimized in the clean room. However, considering that this is a flow cytometry-based isolation protocol, we built in some flexibility for adjusting sort gates and flow rates to mitigate donor-to-donor variability, sample or staining variability, as well as quality of cells on the day of sorting. Moreover, the CGX10 software and the protocol has built-in reference plots for guiding gating strategies. Using the same lot of donor cells as in PD runs, 2 independent CD4 naive T-cell sort runs were executed in the GMP clean room by different sets of trained operators. The flow plots here demonstrate the sort gating strategy. We first gated for lymphocytes and then gated for CD4 positive cells within the lymphocytes. Within the CD4s, we then gated for CD62L and CD45RA double positive cells. These are the CD4 naive T cells as highlighted in this red box. As you can see between the pre-sort and post-sort samples, the sorting by CGX10 highly enriched for the target cells, which are positive for CD4, CD62L, and CD45RA. When we compare the results from the 2 GMP runs to that of the PD run, we achieved greater than 95% sort purity in all the runs, and the results from the GMP run are highly comparable to that of the PD run. Moreover, cells maintained high viability post-sorting, and the results are comparable between PD run and the GMP runs. To summarize the independent operability assessment of CGX10 in GMP, we have demonstrated that trained operators were able to execute the batch records for all the 3 protocols independently in the clean room. CGX10 has an easy-to-follow software and a user-friendly interface. It provides stepwise guidance for kit installation and protocol execution. Protocols can be locked on the CGX10 before moving into GMP, and this would minimize manipulation of gating strategy in the clean room. However, flexibility can be built into the batch records for sort gate and flow rate adjustments to accommodate for donor-to-donor variability and variabilities in sample preparation. Moreover, CGX10 software has inbuilt reference plots for guidance for the adjustments. Another cool feature is that there is operator switch function, which can be used to accommodate long sort times. Moreover, CGX10 generates signed sort reports that captures all user operations. To summarize this work, we have now shown that CGX10 is a GMP-ready instrument that can be integrated into CGT manufacturing in a GMP clean room. Thank you.
Thank you, Manoja. In summary, the CGX10 Cell Isolation System from Sony is ideal for high-speed isolation of highly pure and viable T cells, stem cells, and NK cells, exhibiting different immunological markers. With its GMP-friendly instrument design, single-use kits, and software, the instrument is a first mover in the cell and gene therapy cell isolation space. It enables users to navigate the cell and gene therapy manufacturing workflows with confidence. Thank you, everyone, for attending this presentation. If you want to learn more on how the CGX10 Cell Isolation System can add value to your current cell and gene therapy manufacturing workflow, please visit our website at www.sonybiotechnology.com.
Thank you, Jerry and Manoja, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question, it looks like, is for Jerry. And that question says, how many lasers and how many parameters can be used with the CGX10 system?
Well, thank you, Cassie. So the system has up to 4 lasers and can detect 8 fluorescent parameters as well as forward and backscatter, so 10 in total.
Wonderful, thank you. Our next question that we have here looks like is for Manoja. That question says, please comment on viability of cells after CGX10 sorting.
Hi, CGX10 sorting is very gentle on the cells. We have routinely seen that the viability is actually enhanced after cell sorting. And the sorted cells expanded really well in culture.
Wonderful, thank you, Manoja. All right, I think this one goes back to you, Jerry. This question asks, what are the regulatory and quality support documentation provided by the CGX10?
Let's see. So we offer certificate of analysis and sterility information, as well as certificate of origin. Those are typically for the consumables, as well as an SDS. We also offer certificate of conformity on the system itself, and then device master files for the US, as well as product information files for EU and Japan.
Great, thank you. All right, our next question here looks like is for Manoja. It says, which criteria did you follow for particulate count for GMP use? Manoja, were you muted, I think? Sorry.
Hello. So we used the EU GMP Annex 1 acceptance criteria for the particulate counting. And the CGX10 passed the criteria with a huge margin. The particles that were generated were less than 10% of the acceptance criteria.
Okay, great, thank you. All right, our next question here goes back to you, Jerry. That question says, what is the difference in software operation in CGX10 PD mode versus OP mode?
Well, the PD mode, or process development mode, really gives you a lot of flexibility to set up your templates, perform compensation, get your worksheet, all basically in preparation of preparing your SOP for the OP mode. So it's really meant to be handled by somebody with some flow cytometry experience that can, really kind of set up and get the assay ready on the cytometer. However, during the OP mode, or the operator mode, that one simply just uses the SOP created in the process development mode, and is a very linear, very guided workflow that is really designed for the manufacturing process, and really lowers the bar so that the operator doesn't have to have a lot of flow experience. They can just follow a step-by-step checklist through the software and verify a few points, and then they're ready to sort with very, very little training necessary.
Great, thank you, Jerry. All right, we have another question here for you, Manoja. This question asks, what are the routine QC measures recommended for the CGX10?
So for CGX10, because it's a flow cytometry-based instrument, the vendor recommends a monthly QC run using beads. And it's performed during IOQ, but a monthly run is recommended to make sure that the lasers are in alignment and the fluorescence detection parameters and the performance is not degraded over time. And within our GMP facility, we recommend that this QC run be performed every time the instrument is moved between the clean rooms. It's a very sleek mobile instrument, but any small movements can have impact on the laser alignment. So just to be as a quality check, the QC run is recommended every time it's moved.
Fantastic, thank you. And it looks like we have time for one more question here. That question we're going to pass over to you, Jerry, for the last one. This asks, does the CGX10 provide IQ or OQ and vendor qualification support?
Yes, it does.
Great, thank you. I want to thank you again for your time today, Jerry and Manoja. We have one final poll question that's going to pop up on the screen for us. So please, if you would take a moment to record your responses. We want to thank you for your time and your important research. We would also like to thank LabRoots and our sponsor, Sony Biotechnology, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during our on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.